Thank you for all of you for being here today. And today I would like to give a short overview actually of adverse outcome pathway development illustrated by two use cases. And yeah, can you hear me well?
Yeah.
So for those of you, for those of you who are just not familiar with the concept, I'll just start with a short introduction. What is AOP? It's an analytical construct that describes a sequential chain of causally linked events at different levels of biological organization, and that can lead to an adverse health or ecotoxicological effect. And why it's so important? Because AOP is aimed to be used as a framework to support risk assessment based on mechanistic reasoning. There are different stages of development of AOP. We can have computationally predicted AOPs, putative AOPs, formal and quantitative AOPs. So the more we go towards quantitative AOPs, we have increased confidence, but limited amount of AOPs developed. So today, actually, in this presentation, I'll give two examples of AOP, which is more what are the advances towards computationally predicted AOPs and formal AOPs. And this is more connected to the work we have done within Edelweiss Connect. So the first part is actually focusing on skin sensitization as an endpoint. And this is skin sensitization is an allergic response to a substance after skin contact, and the clinical manifestation is allergic contact dermatitis. Normally there are two phases, the induction phase to the sensitizer, and then the elicitation phase to elicitation of response. During the first phase, what's more specific is that you have the penetration through stratum corneum of the sensitizer, possible metabolism, formation of electrophilic substance, and irreversible binding to proteins, and finally formation of a HOP10 protein complex, which can be then presented either by the dendritic cells or active response in keratinocytes to the lymph nodes, where it would trigger T cell activation, proliferation of T memory C cells, and eventually in secretion of inflammatory responses, cytokines release, cytotoxic T cells. So the whole thing is actually the first response, the first contact with a certain sensitizer, and once we have a second contact, this is happening much faster. So normally for skin sensitization, we can claim that something is sensitizer only if there is evidences in humans or after appropriate animal tests. That's the definition. But of course, there is more and more push towards reduction, replacement, and refinement of animal experimentation, because those tests are most of the time causing a lot of distress, and almost all of the legislations are oriented towards replacement of animal experimentation. But where do we need nowadays? I'm sorry for the people who are, it's more European based. This is more legislation requirements for Europe. In Europe, actually, we need still information on skin sensitization as an endpoint in REACH, and biocides, pesticides, CLP, and cosmetics. In the cosmetics, we know that we have ban on animal experimentation, so we really need to look for alternatives. And there come in place actually the concept of adverse outcome pathway. For those of you who are not familiar, there is a adverse outcome pathway 40, in which actually the whole process of skin sensitization is described, starting with molecular initiating event, which is the covalent interaction with screen proteins, going through key event 1, which is the processing by dendritic cells, through key event 2, reacting and activation of response in keratinocytes, and finally T-cell activation and proliferation, and the adverse outcome pathway. The idea is that actually those different steps can be targeted by in vitro assays, and as such information can be gathered and conclusions can be drawn on the sensitizing potential of the new chemical entity which is tested. And from the OECD, there are several assays and which are actually accepted. This is the direct peptide reactivity assay, HCLAT, and keratinosense. They use different test systems. The direct peptide reactivity assay is the test system is a synthetic heptapeptide, and the endpoint is percentage of cysteine or lysine peptide remaining in the DPRA assay. In keratinosense assays, we have a transpected cell line, and here the endpoints are the concentration, which are causing 1.5 or 3 time induction of NRF2 dependent luciferase activity. And the third, in the HCLAT assay, we have human monocytic cell line and here actually we have as an endpoint concentration which are causing 150 or 200% induction of cell surface activation marker CD86 and CD54 respectively. And the reduction of cell viability is monitored as well. So what do we do actually when we have 3 different assays? They are targeting actually the different, as I showed, different parts of the AOP. So we can either go for majority voting from the outcome of the 3 assays and then just decide whether something is sensitized or not. We can implement multiple linear regression model. The goal is actually to see the relationship between dependent and independent variables. And the third one, which is actually more developed within our company, is the Bayesian network approach. This was actually an approach which was first described and explored by Joanna Jaworska in 2015 in a paper. And the approach actually consists of combination of inputs coming from physicochemical properties, in silico potency prediction, and in the 3 in vitro test results. And a Bayesian network is formed, which would actually lead to prediction of probability, whether something is a non-sensitizer, weak sensitizer, moderate, or strong sensitizer. So what we have actually developed within our company is an application which is gathering this information from the 3 assays and is applying actually the different methodologies, a Bayesian network methodology, multiple regression, and 2 out of 3 voting, so majority voting. And actually, if we compare it with the Joanna Jaworska's paper, it's on the same number of chemicals, so the same data set, 107 chemicals. The network structure of the Bayesian network is the same as she has developed it. And we try to be as close as possible to what's described there, but of course, some models had to be built in-house. And as you can see here, the comparison between the 2 models that shows that the accuracy of prediction is quite close. And why we would say that the Bayesian network approach provides it's kind of better than the other 2, the majority voting and the linear regression. It's mostly actually because also because of all the reasons mentioned here actually. And one thing that needs to be taken into account is that here we can say not only that it's a sensitizer or not, but we can also predict whether it's a weak or weak sensitizer, so we can have some kind of a potency of the sensitization. And the solution can be used also quite easily and fast. So for those of you who are interested, here is just a link to the application. We can just go and select the molecule. This is how the tool looks like. You can try it out and then select the molecule. In this case, I just started actually with octanotriol, and you can say make a prediction and decide whether, and explore the different ways. I've just chosen a few cases where one is a non-sensitizer, the other one is a sensitizer. Interestingly, of course, there are also other tests which can be included in such an approach, like the census. So far, this test is not included in the application as such, but it seems to be a quite interesting test with quite high accuracy and sensitivity. And in this case, for example, it's actually made better prediction than the other 3 cases. So for those of you who are interested, can just go and have a look a bit more in the application. So the second part of this presentation is actually on computationally predicted AOPs, and this work is part of a Marie Curie action. And as I said, this is more or less what should be the structure of an AOP gathering evidence is a different level of biological organization. So the general idea behind this project was that we can use publicly available information on different data sources, namely ToxCast, comparative toxicogenomics database, Reactome, TGGates, in order to gather information from them, extract information, and try to see what is the relationships between all these big sources of rich information. So from ToxCast, we extracted the chemical gene relationships, and from comparative toxicogenomics, chemical genes, chemical disease relationships, Reactome information mostly on gene pathways, and TGGates, chemical genes and chemical pathologists information. So this is more how actually we created an automated workflow, although currently in the current form, it's, I must say, more semi-automated workflow because it's just a series of R scripts. But the goal maybe in the future is to create a tool and application on top. So far, it's just a series of scripts. But today in this presentation, I'll illustrate the usefulness of this actually automated approach, semi-automated approach, by one use case for non-genotoxic induced hepatocellular carcinoma. So in brief, we have created 2 modules. This is the first module is information gathered from ToxCast and CTD, and by actually executing a association mining algorithm on all the data and present in this 2 databases, we managed to create our own database on gene pathway disease relationship, which we could from here on query for different information, starting with a disease of interest. For the second module, we actually implemented it mostly because the information coming from the first module was quite not so specific. So the second module was integrated mostly to make it more focused and more specific and customized. And this is the part where so far there is still some manual input needed once if you decide to try out the R scripts later on. So in the second module, we have the genomics data, histopathology, biochemical, hematology data. And yeah, actually by applying different mechanisms of pre-filtering, we were able to extract actually the most specific information at the different level of biological organization associated with the certain disease of interest. To make it more clear, because I'm totally aware that so far most probably you're still confused with this quite complex figure, is I'll just go through use case, and I hope that it will become more clear to you. So the use case is non-genotoxic induced hepatocellular carcinoma. It's quite ambitious use case, but also quite interesting as well. So for those of you who are not familiar, I'll try it with a short introduction. I mean, actually for the causes, triggers that can cause cancer. One of them are chemicals, and you can have genotoxic and non-genotoxic chemicals. So the genotoxic carcinogens are changing quantitatively and qualitatively the information and the genetic information, and as such, they can cause cancer, whereas non-genotoxic carcinogens, they don't bind to the DNA and are not mutagenic, and they still can cause cancer. Nowadays, to identify whether something is a carcinogen or not, we still rely on the 2-year rodent carcinogenicity bioassay, which requires 2 species, 3 to 5 treatment periods, control group, at least 50 animals of each sex, and for the duration of 2 years or the lifespan of the animal. So it's really a huge amount of animals, so alternatives are very much wanted. But the problem is not so easy actually to be solved. So in vitro, what do we have? We have the cell transformation assay. We have different types. I just gave an example, the SHE assay or the BHAS assay, and both actually have similar principle. You have exposure for a certain amount of days, then colony scoring. The claims is that we have quite a high concordance with the in vivo rodent carcinogenicity data, and that they're able to detect also non-genotoxic carcinogens. The problems, however, is that the mechanisms behind are not really fully understood, so we are dealing with a bit of a black box, and we have really subjectivity of scoring. So these are 2 main problems which still have to be addressed, and that's why this assay is not really gaining, I must say, not so much popularity. The other in vitro assays which are available are more focusing only on genotoxicity. We have the regulatory accepted assays, more going mostly towards execution of only 2 test battery, MS test and micronucleus. The claim is that we have really concordance with the positive in vivo assay, which is quite high. The potential problem is that genotoxicity doesn't always mean carcinogenicity, because you can have repair mechanisms and so on, and concordance with the in vivo, with the negative in vivo assay is quite low. So, do we understand the question, the non-genotoxic carcinogens? This is just a schematic representation of some literature extracted data. On this side are the chemical initiators which act through non-genotoxic mechanisms, which are known to act through the non-genotoxic mechanism, and this is how they're described in literature. And there are some AOPs, formal AOPs described on individual mechanisms, but as you can see, the picture is much more complex. And of course, the question comes: are there any actually mechanisms which are more important than the others, and that are major players which are common for all of these chemical initiators acting through non-genotoxic mechanisms? So that's what we actually try to see by executing our automated workflow. So the first part of the workflow was, we started with the module 1. So it was the database on gene pathway disease associations that I mentioned before. We just filtered for hepatocellular carcinoma because non-genotoxic induced hepatocellular carcinoma was not present as a terminology. So we could only filter for hepatocellular carcinoma. The terminologies for disease we take as from the dictionary present in comparative toxicogenomics database. So what we ended up is with around 250 genes involved in 25 major pathways. And this is combined genotoxic and non-genotoxic exposure. And as you can see, it's quite general outcome, I must say. So it's difficult to say which one is more important than the other. So if you try actually to make it a little bit clearer that what the picture looks like, you can have some different modules like transports and DNA replication and extracellular matrix organization, which are identified there, but it's still quite complex picture. So we had to find a way to refine it. So that's why we introduced the second module. And in this case, in order to focus more towards non-genotoxic induced hepatocellular carcinoma, we selected as a beginning set of reference chemicals, which were split into 3 groups: genotoxic carcinogens, non-genotoxic carcinogens, and non-carcinogens as a negative, negative control. So we explored the transcriptomic samples, in vivo data only, liver only. So it's a bit a restriction of this workflow in particular. And then the hematology, biochemical data, and then histopathology. So on the in vivo gene expression data, we started with all doses, and in this particular database, we had high, middle, and low doses, and we had a lot of exposure time points starting from 29 days, 15, 8, 4, 1, 9, 3, 1 hours. The preliminary analysis showed that actually the low doses as well as the time points lower than 8 days kind of showed very inconsistent vague response, I must say, and limited response. Some of the response we just directly saw it at the more extensive time points, like 15 days, 8 days. So for the sake of more simplistic presentation, we actually focus more on just high and middle dose, 29, 15 days, and sometimes 8 days as well. So what our workflow was is that we actually directly extracted the data on fold changes and made preselection according to fold change and p-value, and we continued only with the genes which are deregulated and present in minimum 3 out of 10 chemicals in each toxic group. So to have actually, the idea was to have genes which are rather toxic group specific, not individual chemicals specific. Then we took the outcome from module 1 on hepatocellular carcinoma associated genes, and then the outcome at the gene level from module 2 for non-genotoxic specific hepatocellular carcinoma associated genes, and then we continued from there on only with the overlap between time points and doses, and explored all these genes, actually all this strategy at the different doses and time points. And this is just a Venn diagram showing the genes which found to be in common, and after going all these pre-selection criteria. So then we decided actually that we need to go in each individual gene and explore the data behind, because bioinformatics can be sometimes interesting. So what we saw is that actually for some of the genes, although they were selected, the problem was that there was some missing values. Like here, the gray spots were missing values, so those we had to exclude. Then we saw that the initial trend, there was quite a nice trend of grouping of non-genotoxic carcinogens according to this selected criteria, which was promising. And that actually the outcome was quite similar for 15 days middle doses. So that was quite promising as well. So what we did later on is we refined the set of genes for which there was missing data. So these are the gray grayed out genes. So we removed them for further analysis, and then starting exploring the individual genes, how they were deregulated as a function of time. So we have here 29 days of exposure, 15 days, and 8 days. And then we decided to put some kind of a pre-selection criteria, whether we see a good or bad, in a way, biomarker. So in blue, you can see here, in blue, the bars are denoting non-genotoxic induced hepatitis, non-genotoxic carcinogens, the group of non-genotoxic carcinogens. And the idea was that this group was supposed to be differentially expressed in comparison with the other two groups, the green bars and the orange bars. And yeah, actually by going through this criteria, we saw that, and this is just an example for cited 2, we see that it's downregulated, and this might be associated with increased apoptosis and so on. So by applying all these criteria to the selected genes, we actually went to approximately 30 genes to something like 10, 11 genes, which were of a good quality and were actually genes we thought are interesting to explore further. So what was the next task is to see, are those genes connected in any way? And we were able to identify actually 3 major processes in which they were involved. So the green ones are the ones from the lists that we have selected from the workflow. The gray ones were selected from the workflow, but for them there was missing data, so we couldn't really conclude anything specific for them. And then the transparent white ones are the ones that are just found as the connector genes. So what we could see is that we have 2 main processes which are interesting. This is cell cycle proliferation and endoplasmic reticulum stress. The third process in both was on apoptosis, but there was only 1 gene which was selected. So the next thing we did is we went through the hematology biochemical data present in TG gates and selected again the same doses and time of the exposure, and applied similar criteria actually as at the gene level. So the selection of parameters which are deregulated and present at least 3 out of 10 chemicals per toxic group. Again, then after this preselection criteria, we decided to see actually what's the reality and how indeed these are expressed by making a dual clustering, like heat map. And what we could see is that actually there was only 1 parameter which seemed of interest, differentially expressed. It was ILP, and it seems to be mostly upregulated following non-genotoxic exposure, but not genotoxic and non-carcinogenic group. So then we went into exploring a bit, in the individual classes. And the last set of data that we were interested in was the in vivo histopathology data. And there we focused mostly on severe and moderate damage, which as identified by the histopathological findings described in TG gates. And then we went for non-genotoxic specific histopathologies only, so ones which were present in the non-genotoxic group. And there, what we saw is that we have a lot of degeneration, alteration, proliferation, which are pointing. I mean, it was interesting to see, but of course now the question was how we can combine all this information into something meaningful and more AOP-like. We don't claim that this is a AOP, but I would say a starting point towards maybe generation of AOP in this direction. And of course, it needs to be further explored and validated. But yeah, this is how, in principle, we organized all these findings. At the molecular level, we put the different genes of interest as identified, and then the three major processes in which they were involved: increased cell cycle proliferation, endoplasmic reticulum stress, and actually resealing of plasma membrane disruption. And then we tried to connect them with the finding at the tissue level, which could explain quite nicely some of the findings. And then at the organ level, here we had the increase, actually, the hyperplasia and necrotic level, which eventually we assume might, well, it is described in literature, they lead to non-genotoxic induced hepatocellular carcinoma. So what we could say is that this tool could be a valuable, actually, this could be a valuable tool to be freely exploited by the scientific community and to provide the links between events in a very easy and unbiased and fast approach. It allows actually for a use of public information that's hanging in there, and actually is very valuable and interesting to see how it can be, what's the additional information that can be added. And it can serve as a starting point actually for the experts to go in there and see what could be indeed major processes which can be triggers of development of certain disease. So towards specifically for non-genotoxic induced cancer, well, the alternatives are currently not very helpful, and maybe the AOP concept is the right way to go forward. Normally, this is linked to the code where it's in GitHub, where the series of R codes are present. So if any of you are interested, you can just go and have a look. But as I said, it's not fully automated, although it would be nice to, but it can be quite useful in the present form as well, and it's free, so you can maybe go in there and play a bit with the code if interested. So both of these projects actually were subsidized, funded by the advanced projects, which is Molecular Reaction and OpenRiskNet new project. Thank you for your attention.